Thank you very much, Professor Costa. Martin is Costa. I'm always confused about these different names in Spain. And thank you very much to Dr. Marisa Vidal from Nestle for the invitation to come to this really beautiful place to talk about human milk oligosaccharides and the latest developments. I thought a few days ago that I'd just do a literature search, had a look at PubMed, what's extremely new, and I'll show you two slides, the first two slides, so you get an impression why these HMOs might be really important to investigate further. Okay, now here's the first one, published on Monday, that HMOs obviously have an effect on necrotizing enterocolitis, at least in an animal model. It needs to be shown that these can be translated into humans. And then there is another one demonstrating that human milk oligosaccharides and not prebiotic oligosaccharides have an effect on very special cells like T cells, immune cells, in neonatal pigs infected with rotavirus. And the newest advancement are that we finally, after many, many years, we do have human studies. There are two cohorts from Abbott and from Nestle, and you will hear more about these studies from Marisa Vidal later on. So we have been waiting really since 30, 40, 50 years to be at this stage now. And you might wonder why these HMOs are of so great interest at the moment. These are not new components found in milk the last 10 years, but it started a long, long time ago when Richard Kunz, he received the Nobel Prize for chemistry in 1937, and Georgi, they realized Kunz was identifying those components we are talking today already in the 1950s, and together with Georgi, you all know the very well-known pediatrician, and they investigated together and found that in human milk there are growth factors, especially for bifidobacteria. And they were not only looking at structures, but they realized that the effects are due to the production of lactic acid and acetic acids, exactly the same what we call today the prebiotic effect. Now, meanwhile, it took quite a while because there were no methods available to analyze these components. We know much more, and I come back to newer dates. We know today that in human milk, we have about 5 to 15 gram oligosaccharides, which is a huge amount. And these are not present, these are not in bovine milk, or only a few in some traces. So that means HMOs are, from a compositional point of view, the largest gap between human milk and infant formula, if you want. Now, the biggest breakthrough occurred when some companies have been able to produce at least a few of the smaller oligosaccharides. And now we are at the beginning of a very new era. It will last 10, 15 years, where we are able to do human clinical studies. And it's really very exciting. Just one slide about potential functions. The hottest topic is the influence of HMOs on the microbial composition. And there are quite a lot of in vitro data and animal data showing that HMOs have an influence especially on special types of bifidobacteria. But there are other effects as well. For example, their anti-preventive effect, preventing certain pathogens for adhering to the epithelial cells here. We and many others have shown that there is an influence on gut maturation, on cell cycling, on signaling and so on differentiation and immune effects. And that's, together with the microbiota, that's the link to certain gastrointestinal diseases. And the interesting point, or one interesting point also, is that obviously the Lewis and blood group secretor status of the mother has an influence on the milk composition and then most likely an influence on certain diseases. I will come back to this later. I will focus on HMO structures a little bit because you will face those structures in the next years. And then I come to the main point, is some metabolic aspects, and then I close with some very unique influence of the Lewis blood group and secretor status, which we need to pay attention to in the future. Now, I don't go into detail, but it is rather simple. We have always these 5 monosaccharides, glucose, galactose, which form lactose. Then we have an amino sugar, the sialic acid and acidic monosaccharides, and the neutral monosaccharides, which is fucose. And these monosaccharides can be attached to the lactose backbone, and then you get components, for example, 2-fucosyl-lactose, which is already added to an infant formula, where the fucose is linked to a specific position on the galactose or on the glucose. Or if you add an acidic sugar like sialic acid, you get 3-sialylactose, 6-sialylactose, depending very much on the linkage. The linkage is important because talking about the prevention of adhesion of pathogens, it's very important if it's a 2-3 or 2-6 or 1-2 linkage. Now, finally, the last slide at the moment. These are the core structures, lacto and tetraose, called LNT and LNNT. The difference is only here on this position. And from these backbones, a whole variety of components are produced. And what we can say at the moment is these HMOs are very unique. They have special functions, obviously, and an unusual metabolism, and they cannot be compared to any prebiotic on the market at the moment. And I'll show you in one more slide what that means and why we cannot compare them. These are human milk oligosaccharides, fucose, lactose, and so on. And if you compare these with fructooligosaccharides, you will see in these components you have only glucose, then fucose, not present in human milk. If you take galactosylated oligosaccharides, it's the same picture. You have glucose, galactose, and so on. And again, those monosaccharides and especially the linkages are not to compare with HMOs. Back to HMOs, the question is, what is their metabolism? It is stated, and we believe they resist digestion, they reach the colon intact, and there they can be used by the microbiota. But we are talking about 150, 200 components which have been identified, and the picture is not as simple as it seems to be. We assume that if HMOs are used by the microbiota, then we shouldn't find those in fecal samples. And then the question is the effect specific or rather unspecific? Does the effect depend whether we feed term or preterm milk, or does the Lewis blood group and secretor status plays a role? That's the picture at the moment. We know quite a lot, I would say, about the HMO intake, the amount, the pattern. We know that the microbiota is using maybe most of it, but we don't know too much about the amount which is excreted with feces, whether there is a certain pattern, and we don't know too much about the urinary excretion. There was an earlier very good paper about 30 years ago showing obviously that smaller oligosaccharides are excreted with urine, which is unusual because these are not monosaccharides. We thought a few years ago, how could one be able to label these HMOs to be able to follow their metabolic fate in infants? And then we started to apply C13 labeled galactose to lactating mothers, and we had hope that this galactose is transported through the liver into the mammary gland, and we thought hopefully we will reach an enrichment of the lactose or maybe even of oligosaccharides. And I could talk for hours about these studies. To make it short, it really happened. We have been able to label those oligosaccharides and follow their metabolic fate. Now, meanwhile, we have other studies done without labeling, but what is important, because we get this increasing number of publications. Some investigate, some address HMOs in milk, some in fecal samples, some in urine, but we really need to, in one study, we need to use all these 3 biological samples to relate to each other. I'd like to show you why. Now, we have this study with Professor Martinez Costa and Dr. Collado, which I really very much appreciate. In this study, we showed there is no difference between term and preterm milk, for example, although many other publications report those differences. Now, the reason is, if we then separate the same cohort into secretor and non-secretor status, you will get this picture here. You see large differences between secretor and non-secretor. Here, it's due to differences in the neutral oligosaccharides. And as you will see in a few minutes, it's not only the total amount which is different, but it's also the pattern which is quite different. And the question is it important to know whether an infant receives secretor or non-secretor specific HMOs? Now, what does that mean? We all, independent of milk and infants and fecal samples and so on, we all belong to secretors or non-secretors. In secretors, 80% roughly in Europe, we have the equipment for certain genes, or for certain proteins, for certain enzymes, which are able to attach fucose in alpha-1,2 position through a specific enzyme on cell surfaces, on glycoconjugates, on erythrocytes, on epithelial cell surfaces, and so on. And those specific components are also secreted within milk. In 20% of us, we don't have these special enzymes, but we have other ones. And these are then called non-secretor. In these non-secretors, we get a different pattern in human milk as well, which I would like to show you. The interesting point is that because the secretor and non-secretor status is discussed as having most likely an influence on various gastrointestinal diseases, like morbus Crohn, rotavirus infections, and so on. And there are quite some data, at least finding associations between diseases and the fucosylation status. That means, for example, Crohn's disease. People have a high risk if they belong to non-secretor status, or with regard to norovirus infections. If you are non-secretor, you are almost completely protected from getting an infection. There are some data on Campylobacter and so on, which prefer the reactions with fucosylated components. There are associations reported about mortality, morbidity, and so on in low and non-secretor infants. And finally, there's a publication from David Mills, very well-known microbiologist, where he finds a relation between the food status and the microbial composition. Now, to come back to human milk, the question is it important to pay attention to these different status? And we know that the neutral oligosaccharides, not the acidic ones, are influenced by the secretor and non-secretor activity. What does that mean to the milk pattern? this looks, what I mentioned before, completely different. For example, you have two samples which are from secretor mothers. And you can see here, these green areas here is LNT and LNNT. And then we have two fucosylactose here, lactodifucotetraose, LNFP1. That's just one simple LNT fucosylated on a certain position. You get this pattern, and then you have a few acidic components, and it's roughly the same here, neutral core structures, 2FL, LNFP1. So these specific structures, they comprise 60 to even 80% of all HMOs, which is a huge amount. In non-secretor milk, 20% of all women, roughly, the picture is completely different. You see here more core structures, but no fucosylated components, no alpha-1,2 fucosylated components. But there are other fucosylated structures, like this one, LAFP2, lacto-N-defucohexose 2, this one, which we find here, but we don't find those in the other group. Very interesting, and we need to do studies to show what effects are caused by these different patterns. Now, just a few words to fecal excretion. There are quite some reports showing that, for example, there is a gradual decrease in HMO excretion during time, a large amount at the beginning, after birth, and then after 2, 3, 4, 5 months, a low amount, and so on. We found in our studies that the picture is rather very complex. Sometimes, in some samples, we find at the beginning huge amounts of HMOs. In some other samples, no HMOs at all, which then would mean that they are most likely used by the microbiota. If you then compare, let's say, 3 weeks after birth with 3 months after birth, sometimes at the beginning, complex pattern, after 3 weeks or 3 months, no oligosaccharides, only a few, and in other samples, it's just the opposite. No HMOs at the beginning, quite a lot at the end. Indicating that obviously, and it's most likely that the microbiota is responsible for this picture. We also find degradation products, modifications of HMOs, and so on. The next question is, what happens to urine? And I show you this next slide, not going into detail, but if we compare secretor milk and non-secretor milk, and we see here the different a few different HMOs from different mothers here. Sorry. If we then compare the urinary composition, you find exactly the same pattern we had in the milk, which is intriguing because, according to textbooks, what we know is that only monosaccharides gets absorbed in the intestine. And now, all of a sudden, we find large structures with 6, 8, 10, 12 monosaccharides still attached, intact, excreted with urine. Which means that these oligosaccharides are absorbed and circulate in the blood, most likely able to influence the various processes before they are finally excreted. And because in our stable isotope studies, we weighed the infants at each suckling period, we knew the amount of milk they received. We have been able to really quantitate the amount of single HMOs. And what we found is that 50 to 100 milligram LNT are excreted with urine. That's a huge amount, considering that these components may have functions, may have effects similar to a drug. With regard to fecal excretions, I mentioned that the pattern is not that obvious as some reports describe. Often LNT is considered to be absent in fecal samples. We didn't find this picture because if there are HMOs, then always LNT is available, which means it cannot have been used for the microbiota. Now, I'd like to conclude and give you some basic principle messages. I think it's clear, we know this since a long time, HMOs are very unique components and we cannot compare those with any others. It's a very complex structural variety. We will not be able to mimic infant formula with all HMOs, which is not necessary, because I showed you that even the smaller ones, they have unique structures. And this structural variety, they predispose them to various functions. And now, yeah, we are just talking about maybe 3, 5, 7 components which comprise the majority of these structures, and that's what we should focus on, in my opinion. Potential functions relate to gastrointestinal health, to inflammation, infection, immune system, and so on. And finally, the last point, because we get an increasing number of publications, I really believe that we should be very careful with regard to the interpretation of the results. Thank you very much.